Hi, everyone. Today we're going to be talking about meiosis, which is a process of cell division that all organisms that are involved in sexual reproduction carry out to make reproductive cells, which are also known as gametes. And in animals, those gametes are sperm and egg cells, which join together when fertilization takes place to give rise to a new to give rise to offspring, right? So last week we talked about mitosis, which is involved in growth and repair, and that created two genetically identical cells. Meiosis makes reproductive cells that are all unique. So they're all genetically unique, and we're going to talk about how that happens. So again, meiosis makes gametes. It makes reproductive cells. So in animals, those are sperm and egg cells. And the important thing I want you guys to know is that gametes have half the number of chromosomes. So in humans, um, our cells, our body cells have 46 chromosomes. Okay, that's the full amount of chromosomes. Gametes or reproductive cells only have 23, which is half of 46. So we get 23 chromosomes from our parent, from our father, and we get 23 chromosomes from our mother. Okay. And when we were conceived, the sperm fertilized the egg, and together, that's where we got the 46 chromosomes that are in all our body cells. So, two vocabulary words we're gonna use in this unit are diploid and haploid. And basically, diploid is the full amount of chromosomes um, that exist in body cells. So in humans, all our body cells are diploid. They contain two sets of chromosomes, right? One set from our mother and one set from our father. And that's true for all animals. So diploid cells with the complete set of chromosomes, which include all your body cells, are diploid. So skin cells, muscle cells, bone cells, brain cells, liver cells, all of those are diploid. They all have 46 chromosomes. The only cells in our body that are haploid are our reproductive cells. And haploid means that it refers to a cell with half the number of chromosomes. How do you remember that? Half starts with an H and haploid starts with an H. So gametes, reproductive cells, are always haploid. Body cells, also known as somatic cells, are always diploid. And they have two sets of chromosomes, one set from each parent. During meiosis, you start with a diploid cell. Okay? It divides to create a haploid gamete. Another thing I want to point out before we go into detail about meiosis is that meiosis, unlike mitosis, meiosis has two divisions. Okay? Mitosis only had one division. So with meiosis, the first thing that happens is the pair. Well, let's actually just look at this picture right here. So in this simplified example, we have one cell with two chromosomes. So let's pretend the blue one is the paternal chromosome from the father, and the pink one is the maternal chromosome from the mother. So during the first division in meiosis, known as meiosis 1, those paired chromosomes separate. So here we've got, in that first division, we've got one cell ended up with the paternal chromosome, and the other cell ended up with the maternal chromosome. After this second division, the chromatids, the two parts of that chromosome, separate, and you end up with, there on the bottom of the left right here, we have two daughter cells. Each got one chromatid from that blue chromosome, and here on the right, each daughter cell got one chromatid from this pink chromosome. But even though there's only one chromatid in each of these four daughter cells, these become individual chromosomes. Okay, so we end up with four haploid cells, each with one chromosome. Okay, so our original cell was diploid because it had the full set of chromosomes. After the first division, we ended up with two haploid cells, okay, each with one chromosome. Then those two chromosomes split apart, and you still the cells are still haploid. They still have one chromosome, but each chromosome has one chromatid. All right. So that's meiosis. So diploid contain both sets of chromosomes, one for each parent. Haploid contain one set of chromosomes, which combine during sexual reproduction to form another diploid cell. One other word I wanted to introduce is this word zygote. So when the sperm fertilizes the egg, that fertilized egg is known as a zygote. Okay. And then after a day after fertilization, that starts to divide. Um, and that grows into an embryo and then a fetus, and which develops in the womb of the mother. So let's practice. Let's practice a question that you might see on the homework. So human cells are diploid. That's abbreviated as 2N, and this means that they contain 46 chromosomes because that's 23 pairs, one from each parent. So human body cells are diploid. Human gametes produced by meiosis are haploid, meaning they only contain 23 unpaired chromosomes. So let's look at this practice question. A fly's diploid cells contain 18 chromosomes. How many chromosomes would its gametes contain? So again, we're talking about meiosis, and gametes are sperm and egg. So let's take a look at how we figure this out. So remember that diploid means the full amount of chromosomes. So that if the diploid number is 18, and you have to remember that meiosis creates haploid cells with half the number of the chromosomes. Um, then how many chromosomes would the gametes have? It's going to be half of 18, which is 9. Okay, so 2N again is basically means the diploid number. So you divide 18 by 2 and you get N equals 9. So the haploid number is 9. So how many chromosomes would fertilized fly eggs contain? Basically, you've got a sperm with 9 chromosomes, which fertilize an egg with 9 chromosomes, and you end up with a zygote, aka a fertilized egg, with 18 chromosomes, because 9 plus 9 is 18. All right, guys, so let's look at the different phases of meiosis. So again, there's two different divisions. There's meiosis 1 happens first, and then you have meiosis 2. So just like with mitosis, before meiosis, you have interphase when the cell grows, and if it gets the signal to go ahead and divide, it's going to replicate its DNA, a process known as DNA synthesis or DNA replication. It's going to grow more and prepare for cell division. Then it's going to enter meiosis 1. So again, during interphase, you have a normal diploid cell. Replicates DNA forming. So each chromosome gets another sister chromatid. So it goes from a chromosome with one chromatid to a chromosome with two chromatids. Again, this happens during interphase. During prophase, the chromosomes become visible. So the DNA, basically that loose unwound DNA condenses into that chromosome form that looks kind of like an X, and basically, the one thing that's different about meiosis is that the male and the maternal and paternal chromosomes pair up. So in humans, we've got 23 pairs of chromosomes. Okay. For each pair, we've got one from our mother and one from our father. And they pair up, they match up specifically with the chromosome that's the same size and carries the same genes. Okay. And those are known as homologous chromosomes. So we have 23 pairs of homologous chromosomes, and they join together during prophase 1, and something very important happens. That's known as crossing over. So during crossing over, each homologous chromosome overlaps and exchanges DNA. Okay. So basically, it would be like if you went up to someone, gave them a hug, and you exchanged arms. Okay, just to mix things up. Well, that's what happens during prophase one. Prophase one is arguably the most important phase of meiosis because crossing over occurs. So, what is the result of crossing over? By mixing up your paternal, which means from the father, and your maternal, which means from the mother chromosome, you're creating genetic diversity in these reproductive cells that are eventually going to be made, which is very important. Okay, so we're mixing up the genes by a process called crossing over, which is the exchange of DNA between matching homologous chromosomes. Okay, so again, exchange genes. So again, crossing over occurs during prophase one. Homologous chromosomes pair up. Each homologous pair consists of a maternal chromosome and a paternal chromosome. During crossing over, the pair exchanges DNA, creating new combinations of maternal and paternal genes. This is called genetic recombination. Again, you're recombining different, you're creating different combinations and different chromosomes, which increases genetic diversity in the gamete. So again, notice how you've got this orange chromosome and you've got the red chromosome. You end up with kind of like these mixed up chromosomes with different combinations of genes. And again, the end result is this is why a couple can never have the same. One of the reasons they can never have the same child twice. Um, Again, because there's so many different combinations of genes you can get um, from two individuals, right? So during now that crossing over has occurred, those homologous pairs stay together. Okay, they stay together and they line up in the middle. So just like with metaphase and mitosis, with meiosis, they line up in the middle. But the difference is with meiosis, they're lining up in double file, kind of like you did in kindergarten. You've got each pair lines up in the middle, in basically two at a time. Okay, um, completely random. Again, and also these spindle fibers, these protein fibers connect to the middle of each chromosome, and they're actually what lines them up in the middle. Then during anaphase one, those homologous pairs are pulled apart. Okay, so they're pulled apart from each other. See ya. They go to opposite sides of the cell. Um, and it's completely random on how they line up and get separated. So that creates more genetic diversity. You know, it's like flipping a coin. You have a 50-50 chance of getting either a paternal or a maternal chromosome. It all depends on how they line up um, during during metaphase one. Love, independent assortment. We'll get to that later. Okay, so telophase now again, just like mitosis, telophase one, the nucleus divides, so we end up um, with two nuclei now, and that happens with cytokinesis, which is division of the cytoplasm and division of the splitting of the cell itself. So those happen kind of concurrently, and then cytokinesis meaning at the same time. And cytokinesis just kind of finishes up the first division. Then you have meiosis two begin, and you know there's no interface between meiosis one and meiosis two. Meiosis two just happens kind of right after cytokinesis is done. So at this point, we're going to start meiosis two. Prophase two is much simpler than prophase one. There's no crossing over because there's no pair for each chromosome to match up with. The homologous pairs have already been separated, so now each chromosome is flying solo. Um, at this point, there again, the spindle fibers just forming and attaching to each chromosome. Nuclear, the nucleus disappears, and those you can see the chromosome here, and there's no membrane around them. Metaphase two, they line up in the middle, but this time they're in single file. They're not paired up. And then anaphase two, now it gets to be just like mitosis. So now we've got our sister chromatids for each chromosome getting pulled apart. And telophase, again, that nuclear The nuclear membrane around the now separated chromatids forms around the separated chromatids. You end up with four daughter cells that are genetically unique. So again, let's go back. Um, at the end of meiosis one, you had two daughter cells, right? And at this point, they are already haploid because the homologous pairs are already separated. So in humans, you'd have 23 in each haploid cell at the end of meiosis one. But notice how at the end of meiosis one, the chromosomes still have two chromatids, right? They still look like an X. At the end of meiosis two, the chromosomes look like a 1 or a capital I, right? They only have one chromatid, but they're still considered haploid. So in humans, these cells would still have 23. Now you have gametes. So again, one key difference with meiosis and mitosis is that unlike mitosis, meiosis creates genetic diversity. It creates daughter cells that are genetically different. And that happens due to, for a number of reasons, and two of them are crossing over during prophase one, which is exchange of DNA between homologous pairs, and the law of independent assortment. I don't want to go into too much detail with that, but basically, it's just the random ways that chromosomes line up before they're separated. So basically, there's a 50-50 chance that for each paired, each homologous pair, you could either get the maternal chromosome or you get the paternal. And that's why some people look more like one parent because they get more chromosomes and more genes from that parent. So it's completely random which one you get. And that the different combinations that you get when they line up and separate during meiosis creates genetic diversity because there's so many different combinations of genes and chromosomes that you can get um, during this process. All right. So genetic diversity just means different combinations of genes. And all right, guys. So real quickly, what's things can go wrong during meiosis. Sometimes chromosomes don't separate correctly for whatever reason, and you end up with gametes that either have too few chromosomes or too many. Those homologous pairs might get stuck together during anaphase, and just like this one on the left, we have a daughter cell here with two chromosomes instead of one, and this one got the short end of the stick. It didn't get any chromosomes, so that's called non-disjunction when they don't separate correctly during anaphase one or anaphase two. So this is what should happen. You start off with diploid cell. You end up with four haploid cell. Each has one chromosome. But non-disjunction happened in this one right here. So you have non-disjunction. All right. So what's the possible result of non-disjunction? One possible result is a disease called Down syndrome. Now, before I say that, I want to say that most of the time when non-disjunction occurs, um, that gamete that's created is not going to be able to fertilize an egg, and if it does, that embryo is not going to survive that embryo or fetus is not going to survive the pregnancy. But in some cases, a child can survive with an abnormal number of chromosomes, and one example is Down syndrome, and they only have one extra chromosome, and that's an extra chromosome 21. So instead of having two, they have three of chromosome 21. So, and that's called Down syndrome, aka trisomy 21. Trisomy means three copies of a chromosome. You should only have two. They have three of that pair. So, of that 21st chromosome, they have three copies. So, this is called a karyotype. It's basically a map of a map of a person's chromosomes um, that the doctors can make to look and make sure that a child has the correct number of chromosomes. So this is a karyotype of a Down syndrome person. So this pair right here is the called um, the sex chromosomes. So they determine whether you are male or female, whether you're born male or female. So you can either be XX or XY. If you have two X chromosomes, like this individual, then you are female. If you have a Y chromosome, so if you're X and Y, then you're male. All right. So this is a female with Down syndrome. That's what this karyotype tells me. All right, guys. So let's just compare mitosis and meiosis. Mitosis results in two genetically identical diploid cells. So I can't stress this enough. With mitosis, the chromosome number never changes in humans. For example, when my skin wants to make a new skin cell, it divides by mitosis, and it starts off with 46 chromosomes, and the two daughter cells have 46 chromosomes. That never changes because all the body cells do mitosis and they're always diploid and they're all genetically identical. They have the same DNA in them. Meiosis makes four cells and it makes four haploid cells that have 23 chromosomes. Okay, in humans because they're haploid. Different species have different numbers of chromosomes. So meiosis creates four cells, and they're all genetically different, and they're all haploid with half the number of chromosomes. Right. So mitosis allows an organism's body to grow and replace cells. Meiosis allows sexually reproducing organisms to make gametes or reproductive cells. So in animals, that's sperm and egg cells. All right, guys, we're going to stop there.